All right, let me check if it's recorded. Yeah, cool. Dr. Navid Kashaninejad received his bachelor's and master's degree in mechanical engineering in 2006 and 2008, respectively. Afterward, he completed his PhD study in 2013 at Nanotechnology University, Singapore. During his PhD studies, he mainly focused on the design and fabrication of different microfluidic devices and fundamentally investigated the fluid mechanics of flow through microchannels. In 2016, he was selected as a distinguished scientist from INEF and awarded a full scholarship and research funding to pursue postdoctoral studies at Shanghai University of Technology. He also has over 2 years of experience as a postdoctoral research at Griffith University and the University of Technology, Sydney. Currently, he's a full-time postdoctoral research fellow at Queensland Micro and Nanotechnology Center and work as a team leader in an industrial project in partnership with IMCRC. So Navid, the screen is yours.
Okay, thank you so much, Sadeghdon, for your kind introduction and also inviting me to this talk. So, because I know that we have friends with different backgrounds, and for me, I was actually thinking about how to present this talk, and finally I prepared some slides regarding my own actually background and how I have been actually introduced to this field, and down the way what has happened to me as a PhD student and later after PhD. So what happened? So I think it's not a bad experience to share with you. So because by the way, if you have by any talk, I think if we have a one takeaway message, that would be also great. So you don't have to actually understand, or we don't have to actually learn all the things from the talk, but hopefully you will actually, we will be inspired, or you will find one useful thing from my presentation, and that would be great for me. So let me start here. So you can see my screen.
Yes,
okay, good. So this is the building for Queensland Microtechnology Facility. We call it QMF. So it is located in Griffith University as a part of Queensland Micro and Nanotechnology Center. So basically inside this room, we have a very good clean room for MEMS and specifically for silicon carbide and silicon technology. It is one of the unique facilities that only in Australia is not, only we have this kind of SiC fabrication on silicon. And in the world, I think it's one of the few centers. So it's been developed through years by industrial engagement with some leading companies, and so they have very good equipment for specifically for MEMS. But for our applications, actually we mainly use for microfluidics. We most of the time we don't need that kind of expensive clean room facility. So we have another building next to this one, that's mainly our microfabrication lab and facilities is there. But if you want to do the microelectronics fabrication, so we need this level of clean room. So first, just want to tell you about my PhD studies. When I joined at Nanotechnology University in 2009, I had no actually background in microfluidics. So just because the system for Singapore is a little bit different from at least from Australia. I don't know from other countries. So we have been given the scholarship from Singapore Agency of Science and Technology, and there wasn't any specific project. So once I joined this group, my supervisor told me that we are going to work on microfluidics and go and find the challenges. So it took me a lot of time and more around 1 year. So I read a lots of texts. I had gone through many difficulties. So what I'm going to show you is not something that, that was ready to do. So I will tell you that in retrospect, when I look at these things, it wasn't that bad, It makes me actually more prepared, and this kind of develop my critical thinking. And this is somehow, I think, the philosophy of getting PhD, which is a doctor of philosophy. You need to learn the philosophy of getting your degree. But some people are actually can join a group with already defined project with lots of funding, and they have 2 or 3 postdoc which help them. But it wasn't the case for me. So what I actually first understand from microfluidics was when we have a normal channel like this, and when we decrease the channel height, surface effects become actually very important. So they are no longer negligible. Some of these effects are variation of viscosity, electroosmotic effects, temperature jump, and slip velocity. So when you go through the literatures, you have this kind of effects. So I was very interested in slip velocity. And when I look at the literature, so we have in around 1820s, actually, once they wanted to quantify the no slip boundary conditions, this no slip boundary condition is not something that can be proved experimentally or theoretically. This is something that no slip boundary condition is correct for based on the observation, but you cannot prove it. When you look at the macro channels, or when you go through the gas at that length scale, so you can see that no slip boundary condition is no longer valid. But this is not really important in macro channels. But why it is very important in macro channels, and what we can do with that, is that pressure drop, which is somehow related to the energy that you need to consume to drive the flow, is actually scaled with the inverse of the length scale to power 4. So when you decrease the length and you go to the macro channel macron, for example, 100 macron instead of 2 millimeters, then pressure drops, if you want to have the same flow rate, will be very high. And this is a problem. So you cannot have the same flow rates. So one of these solutions is enhancing the boundary condition at the channel walls. But the problem is, if you have a smooth channels, the slip length is not something that can give you a lot of differences. You need to have a they call it super hydrophobic channels. Super hydrophobic channels, when I look at the literature, so you can see that, I can see, okay. Yeah, when you look at the super hydrophobic microchannel, in the nature, we have naturally occurring super hydrophobic channels. When you look at the lotus leaf, it's some kind of symbol of purity in some cultures, like Chinese culture, because you cannot find any dirt or debris on the surface of a lotus leaf. And when you look at the SEM images, you can see that this actually water is in partial contact with the solid surface, and there are some air gaps in between. So liquid is actually suspended on that air gaps. And this is the case also for the shark skin. So if you look at the whale skin, for example, it is full of the sea creatures, bacteria, but the shark skin is very clean, and shark is one of the fastest swimmers in the sea. One of the reason is actually this kind of more topography of his skin, and the drag is very low. That's why at high speed, shark can manage to be one of the fastest one. And this has inspired lots of things. For example, swimsuit, this kind of swimwear that they have designed basically with this kind of super hydrophobic textile. And those kind of swimmers, the Olympic ones that actually swim very fast, this really helped them to achieve the that kind of a speed with reducing the drag. So I was looking at this field, which is called biomimetics, and there is a very good reference here that I have put here from Professor Bhushan Bharat. He's a professor in one of US universities. So I wanted to join them, but I couldn't, by the way. Yeah, this is a very good review regarding this biomimetics. So how you can be inspired by nature. So super hydrophobic surfaces is one of them. But for doing that, I have already passed 1 year from my PhD. I joined NTU at 2009, so it was almost 2010. So I had to go for my confirmation exam. So what I did was this kind of fast skin, which is inspired by, as I mentioned, shark skin. I bought it, which was commercially available. I use a laser machine to cut that one. And also I cut PMMA with that laser machine. And at the bottom surface, I actually attached this kind of super hydrophobic textile on the PMMA macro channels, and I fabricated 2 macro channels, one with 250 micron, the other with around 1 millimeter height. And I showed that with using this kind of super hydrophobic surface at the bottom of these super hydrophobic macro channels, we can reduce the drag up to 16%. So this was just to show the proof of concept that we can have this kind of sleep using the sleep boundary condition instead of no sleep boundary condition. We can use it in fluid mechanics to have a practical application, which is reducing the drag. And this was very important. So then I actually become familiarized with Prof. Namtran group at NTU. And I started to learn microfabrication. And then I looked at the morphology of the macro pattern surfaces, which already people use it for making super hydrophobic surfaces. So as I mentioned, it wasn't something that suddenly come to my mind. So after doing lots of literature, so I saw that in terms of surface parameters, for example, the height of the pillars, length of the pillars, spacing between the pillars already has been evaluated in the literature. But I introduced another important topographical parameters, which I call that one eccentricity, which you can see in this figure. So eccentricity is the kind of parameters that you can change with macro pattern surfaces. And this hasn't been evaluated in the literature. And I show that by changing the eccentricity of macro pattern surfaces, we can change the contact angle. And this paper was my main paper, which I published in Langmuir in 2012, which was 3 years after my PhD. So again, it took me almost 3 years, but again, this was the fundamental study, not application study. So sometimes if you want to go through the fundamentals, it's not something that happened overnight. So we have even we have some PhD students here that, you know, by the end of their first year, they already published 6 or 7 papers. So basically, it is not possible when you actually want to work on the fundamentals. So basically, I didn't want to work on fundamental. I had to. Anyway, so we published this result, but after that, once you work on the fundamental things, because this is originally yours, then subsequent publication would be much more easier. So we showed another important characteristic of the wetting behavior on macropattern surfaces, which was the anisotropic weighting. So when you look from different angles with these macropattern surfaces with different eccentricities, at every viewing direction, the contact angle was different. So we published that one in Soft Matter. Again, we fabricated, I fabricated this this time, these macro pattern surfaces with the bottom wall, but and I showed that the drag can be reduced up to 40%. So this was also a main finding of the study that I published in Physics of Fluids. And this were actually mainly as the studies regarding the drag reduction of microfluidic that I mentioned in the title. So I showcase the application of microfluidics for using macropattern surfaces as a super hydrophobic surface. So I introduced a new parameters, and then I actually managed to have 3 or 4 I have another publication regarding the analytical modeling. So, but first publication was very difficult because actually you didn't, I didn't know what is the actually would be the novelty first. But once you find that one, and once this becomes your original work, then it would be much more easier. Even I recently published this one from my, in 2020, I had some result regarding the evaporation. And at that time, I actually didn't have time to publish the results. So in 2020, I showed that evaporation of the droplet is also a function of the eccentricity of micropattern surfaces. Again, because it was my own work. So you can think about, you know, any phenomena on micropattern surfaces, and I can actually publish in that field. So basically, you know, once you have the first one, the concept, the fundamental concept, you can find the application. But my biggest chance and it was actually when I was involved in this paper. So it was an invited review from Advanced Drug Delivery Review. So the impact factor was 15. Mainly when I published, for example, my main result in Physics of Fluids is the impact, because it's an engineering journal, it is around 2. But when you suddenly is involved in a project with in a journal with impact factor 15, so you start to think, so it's a actually should be very exciting area. And it was that time that I was familiar, I became familiar with the application of microfluidic for drug delivery and actually the cancer disease modeling. So I read a lots of paper when I wanted to write this review paper with other colleagues. One of them is Syed Ali Musavi. So I assume that you know him now. He's an assistant professor at Mashhad University of Medical Sciences. With Ali, I was also involved. What we did the drug delivery. We categorized the drug delivery at cellular level, tissue level, and organ level. And I became familiar with the concept of organ on a chip, macroneedles for drug delivery, concentration gradient generators, and I understood that what I know about microfluidic is like a pre-requirement for doing this kind of application. But this field is highly multidisciplinary and cross-disciplinary. You need to have a good biologist in your field, in your team, so that you can publish in good journals. But anyway, you need to have also a good knowledge of engineering to design these systems. So as an engineer, I was very fascinated by the design consideration. And I also, this is a very good review paper also that I read about the microfluidic for cancer detection. So microfluidic for cancer detection, this paper nicely categorized them. So you can use them for isolation of circulating tumor cells. There are lots of microfluidic systems. You can use this microfluidics for molecular diagnosis. For example, droplet-based PCR on chip qPCR, lots of different things for molecular diagnosis. Another category is the tumor biology. So you can use it for 3D culture with spheroids, cell migration, and also you can use it for high throughput screening. So for my case, I was very interested in tumor biology and high throughput screening. That's why I started to read more in the literature, and I came across this kind of concept of cancer stem cells. So when you look at the cancer, most of the drugs after actually killing the initially can destroy the tumors, but after 5 years or a period of time, again, this tumor will come back, and this time it would be resistant. One of these reasons can be related to the cancer stem cells, that the normal chemotherapeutic drugs cannot really target cancer stem cells. And it is like you have actually you didn't kill the root. So after some time, this root come back to life. So another video that actually I was really impressed at that time and was really motivated to work in this area was this video that I show you. It is 2 minutes, and it is actually, I think, worth to just watch that. So this one, can you hear the volume of the?
No, unfortunately not.
Okay. Oh, okay. Let me see if I can explain with the. So this is from from European cancer stem cells. So as you can see here, so I will talk. I wanted that person to talk because, yeah. So this is the tumor that is going to develop, and these blue things are cancer stem cells or circulating tumor cells that gain this actually capability to invade from the blood vessel, and they can metastase to other parts of the body. And this is the main reason that cancer is very actually aggressive, and is the reason for death can be related to that. So mainly when we have a normal chemotherapeutic drugs, they target the bulk of the tumors, not the cancer stem cells, because these cancer stem cells are not grow very fast. But after some time, again, they come back, and the tumor will recur. So another approach is we target only cancer stem cells. This approach can kill the cancer stem cells, but won't really destroy the bulk of the tumor. And this is, again, problematic because after some time, they can mutate again and gain this kind of stem cell behavior and aggressiveness. So again, this is not really effective. One effective method that this group are working is the combinational therapy with chemotherapy and targeted cancer stem cells to kill not only the bulk of the tumor, but also the cancer stem cells. And this is the work that they are doing with actually, this is a research institute. They are a very strong group of biologists. So as an engineer, I was looking at this concepts and videos and my background in microfluidics and all these great work that people have done. So what I did in 2016, so there was a 3-year gap in my academic work because yeah, so some kind of family issues. But anyway, I joined Sharif University of Technology and with this concept. So I write my proposal regarding lung tumor on a chip. And mainly at Sharif University, we work on this concept. One of the good example of the actually papers that we had was the work that done mainly by Kashaninejad. Kashaninejad was actually a master student. He was very good, and now I think he's PhD at the university, PhD student at University of Austin. In this review paper, it's been cited, I think, over 90 times since 2018. We characterized, we considered the design consideration of all this kind of asteroid formation for 3D culture of the microfluidic in microfluidic devices by considering, for example, shear stress, cell loss, diameter of the asteroid, and all these parameters. We actually evaluated and categorized different methods for flow pumping, for asteroid fabrication, on-chip asteroid fabrication, off-chip asteroid fabrication. This is a very good, I think, paper for anyone that it is not only a review paper, but also like an educational manuscript that it's really good from engineering point of view, how we can design this system. We have lots of numerical papers in this field, so I skipped those things. One of the work that was the main work of one of our PhD students at Sharif University, now I should call him Dr. Milad Rismanian, was regarding the design and fabrication of one new type of concentration gradient generators. So normally, when you look at the literature for drug delivery, they have, you can categorize it in 4 different types of concentration gradient generator to deliver the drug at different concentrations. But here, I think this is not, again, the 1 or 2 months work. It took him 4 years to come up with this design. So by combination of concentration gradient generator with micromixers and actually some design considerations, we can have a 5th type of a concentration gradient generator, which can be used to deliver the continuous gradient of the reagents, which is a drug. And here you can use it 2 different drugs. For example, those drugs which can be used for chemotherapy and the drug which can be used for cancer stem cell that target cancer stem cells. And not only it can provide the continuous gradient of the combination of these drugs, it can also provide a sample with a millimeter scale. So millimeter scale here imply a tissue. Most of the concentration gradient generator can deliver the drug in a micron size sample, which is a cell. But the system can deliver to a tissue in a millimeter scale, which is really great. And we published this one in the Journal of Flow Chemistry. It's not a bad journal, but to be honest, this work could have been published in at least Nature Biotechnology, but we didn't have, he already used this tumor mouse brain slice, but using the drugs and things like that was too expensive. And also we couldn't really do any kind of good biological studies. Just we showcased, for example, for the two reagents, we used two different fluorescent dyes. Because we didn't have actually those kind of specific drugs for cancer treatment. They were very expensive, and also it needs some good biologists in the team. That's why if you in this field, if you go through this paper, it's from engineering point of view, it's really a good one. But for the application, which is we call it for two reagent on a millimeter size sample, could be on combination of the drugs on tissue. But because we didn't have this kind of biological, we didn't actually, the title is like this, but it was a really good engineering from concentration gradient generator. Okay, then another things that we can do is with nanozymes. So I also had some work with one of our colleagues at Griffith University. He's mainly a chemist and working on analytical chemistry. So we can combine microfluidics with analytical chemistry, mainly for diagnosis. So one group that is now, I think, is a very good topic. We have, I have already evaluated the microfluidic system with nanozymes. Nanozymes is a new group of nanomaterials that they can show enzyme mimicking activities, and combination of nanozymes with electrochemical biosensors is really a synergistic combination for disease biomarker detection. And this is something that I think is a hot topic, and we can develop and combine lots of different microfluidic system with nanozymes for better biomarker detection. This is the work we published in the Cancers Journal. This is with the long non-coding RNA from ovarian tumor. So one of the challenges with this long non-coding RNA, they are really good biomarkers, but the problem is their concentration is very low in a tumor. So maybe in a range of femtomolar or nanomolar. So it's very difficult to detect this kind of biomarker. Another challenge is that the level of expression of this biomarker is different, and you need to have a multiplex system for detect that. So here we showed that by combination with the screen printed gold electrode, which is a kind of electrochemical detection, and magnetic purification, we showed that we can have a limit of detection of this long non-coding RNA for ovarian cancer in the range of 1 femtomolar. So that was also a good example of the microfluidic system that can be used for disease or cancer detection. So here we talk about detection, not treatment. So we also, I think I just don't want to go through many details. This is another example of that we want application of microfluidic for assisted reproductive technology, which is, for example, for sperm sorting, for infertility treatment. This is also one of the review that I really like. So sometimes when you write something that is really good, you even after I wrote this one, I think in 2018, but still when I look at this one and read this one, I like it because of the way I characterize different microfluidic platforms droplet-based microfluidic, continuous flow microfluidics, paper-based microfluidics, and organ-on-chip microfluidics for ART, which is the assisted reproductive technology. And very recently, this year, we managed to find a company to which is based in Sydney and work on infertility treatment. So we wrote an agreement together and start to collaborate on this field. But still, this kind of work, we can develop the microfluidic system, but again, need a very good team of biologists and other infertility expert to actually guide us. It is not, this one cannot be done, by just academic environment. In a good way. Of course, there are many, especially in Canada, they are working on this field. But yeah, this is another application. So for microfluidic for liquid handling, one of the work and that we are actually doing at Griffith University is liquid marble, which is a new class of the digital microfluidics or droplet-based microfluidics. I would like to call this one as a super hydrophobic liquid. So this kind of liquid marble doesn't matter whether you actually put it on a super hydrophobic surface or doesn't matter. The surface is not matter because it's already coated with hydrophobic powders. Once you put it on any substrate, the contact angle is more than 170, almost 180. So it is completely hydrophobic. And we showcase the application of this liquid marble for asteroid formation. So we showed that with if we have a solidified hydrogel and encapsulate this hydrogel inside this liquid marble, not only evaporation rate would be reduced, but also we can obtain very nice asteroids after, I think, 72 hours of incubation. So this is also another application of, this is very simple. So when you look at the liquid marble, the way that you fabricate the liquid marble, it doesn't really need any specific equipment. And because it's simple, it's beautiful. And but still, you should think about, innovative way of enhancing the platform. Anyway, so I think I will finish my talk by acknowledging the people who, lots of people actually has done, have been collaborated with me, has helped me. But among them, I always want to thank my PhD co-supervisor, but he was actually my main supervisor, Professor Nam Trang Nguyen, that I was still fortunate that after in 2013, he also joined Griffith University as a center director. So I'm still fortunate enough to work with him, and this is the kind of life-changing experience. So another advice, my advice for PhD student is, it really doesn't matter what you are working and in which university you are. It really matters who is your PhD supervisor. So this always you will be remembered by your PhD supervisor, and try to actually select your PhD supervisor wisely, because it will really affect your academic life, even your normal life. So in many occasions, so I approached him and asked him about very private matters.
Right. I guess he lost the signal. So we will wait for him to rejoin. And then we will start the discussion part. So, yeah, I couldn't see his profile here. All right, thanks for attending this meeting. We will wait for Navid to be joined, and then we can ask him about the different aspect of microfluidic application, and then we will finish the talk. Hi, Navid.
I was disconnected, right?
Okay, it's fine. So yeah, just.
So did I finish everything, or?
Yeah, you were talking about the acknowledgement from your supervisor. Yeah.
Yes. Okay, so then, yeah, I don't know why I've been disconnected, but anyway, yes, this is just my advice that really try to choose your PhD supervisor wisely. So no matter which university or what field you are working, of course it's matter, but one of the very important things is actually your PhD supervisor. So yeah, that was the main thing.
Great. Thank you, Navid. It was an interesting topic about the microfluidic, and also you explained and showed your academic life and your challenge